Welcome to the Summary of Biology. A good place to start is at the molecular level with the carbon atom and its essential role in life with the carbon cycle. Carbon enters the human body through the foods we eat, which are composed of carbohydrates, lipids, proteins, and nucleic acids. When humans exhale, carbon is released as CO2, which journeys into the atmosphere. Carbon is an element composed of 6 protons, 6 neutrons, and 6 electrons. It forms covalent bonds with other elements and is essential for life on Earth because of its critical role in the carbon cycle. Carbon in the form of CO2 enters the plant through the stoma, located on the underside of the leaf. From there, CO2 is brought to the chloroplast of the cell, which are now able to produce sugars. The Calvin cycle occurs in the thylakoids of the chloroplast. In the Calvin cycle, CO2 is turned into 3-PGA with the help of NADPH and ATP produced in the light-dependent reactions. The 3-PGA is then transformed into G3P, which exits the Calvin cycle and is turned into various types of sugars like glucose, sucrose, starch, and more. The remaining G3P continues through the cycle and is turned into RUBP. After this transformation occurs, the process repeats itself. Glycolysis occurs in the cytosol of the cell. During glycolysis, one glucose molecule is broken up into 2 pyruvate molecules. NAD+ transports electrons and hydrogens away from the glucose molecule to the electron transport chain and turns into NADH. A net of 2 ATP is produced during glycolysis. During pyruvate oxidation, pyruvate has more electrons taken away from it with the help of NAD+. Carbon dioxide is also generated during this process. Acetyl-CoA, a 2-carbon molecule, is formed at the end of pyruvate oxidation. The Krebs cycle occurs in the matrix of the mitochondria. During this process, NAD+ and FAD borrow electrons from acetyl-CoA. CO2 is produced as a byproduct during this process. The final products of the process are NADH, FADH2, 2-ATP, and carbon dioxide. CO2 produced from pyruvate oxidation in the Krebs cycle is released through the stoma back into the atmosphere, waiting to go through photosynthesis and cellular respiration once again. The carbon cycle occurs on a molecular level inside of cells, but how does oxygen get into our cells as carbon dioxide leaves? This happens through cell transport. Simple diffusion is a form of passive transport where small nonpolar molecules diffuse through a semi-permeable membrane without the aid of a protein. Simple diffusion requires no energy because the molecules move with the concentration gradient. Simple diffusion allows molecules like oxygen and carbon dioxide to diffuse through the membrane. Facilitated diffusion is a form of passive transport that moves ions, polar molecules, and large molecules across the plasma membrane with the help of a protein. These molecules move with the concentration gradient and require no ATP. Facilitated diffusion allows molecules like glucose and charged molecules to pass through. The sodium-potassium pump is a specialized protein that pumps 3 sodium ions into the cell for every 2 potassium that are moved out. The sodium-potassium pump requires ATP because it is moving the ions against the concentration gradient. The sodium-potassium pump plays an important role in generating electrical signals in our neurons. Proteins aid in cell transport, but how do these proteins know where to move and what to do? This is coded for in their DNA. In every cell, DNA duplicates itself in a process called DNA replication. The helicase enzyme opens up the DNA strand into 2 separate strands. It travels from the origin of replication through the DNA strand, breaking hydrogen bonds in the process. The primase enzyme lays down an RNA primer along the open strand of DNA. The DNA polymerase enzyme builds the DNA strands. It starts with the primers and matches new nucleotides to the complementary nucleotides on the old strand. DNA polymerase also takes out the primer and replaces it with DNA. The ligase enzyme connects any ends of the DNA fragments that aren't connected. DNA replication occurs during interphase, specifically in synthesis. At the beginning of DNA replication, an enzyme called helicase opens the two strands of DNA from the origin of replication. The helicase breaks weak hydrogen bonds, allowing for the strand to be separated easily. Next, RNA primase lays down RNA strands in preparation for the DNA polymerase enzyme. Without the primer, the DNA polymerase would not be able to lay down DNA and create a new strand. After the primase, DNA polymerase reads the strand in the 3 to 5 direction, meaning it places the nucleotides in the 5 to 3 direction. Each strand is anti-parallel, so the strands are going in opposite directions and are complementary to one another. However, a problem occurs with the DNA polymerase laying down in one direction. While the DNA polymerase following the helicase can continually lay down the nucleotides, the DNA polymerase on the other strand will have to keep going back and forth since it's laying in the opposite direction. This causes this strand to replicate slower and become the lagging strand, as the other strand is the leading strand. Once the DNA polymerase is finished laying down nucleotides, it will replace the primer with the complementary nucleotides. After the DNA polymerase is finished, an enzyme called ligase will run through the DNA strand and connect any DNA fragments that aren't connected. The new strands are semi-conservative, meaning that each strand is made of the original strand and a new DNA strand. The strand will now continue through cell division. DNA replication is a process that occurs during synthesis, which is a stage in cell division or how cells multiply. The cell spends 90% of its time in interphase. Interphase is split up into 3 parts: G1, synthesis, and G2. During G1, the cell grows and builds proteins it needs for cell division. It also takes in nutrients. In synthesis, the cell replicates its DNA, so each chromosome has 2 chromatins. Finally, in G2, the cell grows and builds proteins in preparation for mitosis and cytokinesis. Centrioles are duplicated in G2. In prophase, chromatins are condensed into sister chromatids. Centrioles begin to move towards the opposite end of the cell and start forming spindle fibers. Finally, the nuclear envelope breaks down. During metaphase, the nuclear envelope disappears and the chromosomes line up in a straight line at the center of the cell. The spindle fibers, cast from the centrioles, attach to the chromosomes. During anaphase, the sister chromatids split at the centromere and move towards their respective poles of the cell. In telophase, the spindle fibers dissolve and the chromosomes slowly loosen into chromatin form. The 2 nuclear envelopes begin to reform as the cell readies itself for division by forming the cleavage furrow. During cytokinesis, which is the final stage of cell division, the cell splits itself into 2 daughter cells by expanding the cleavage furrow with the use of microtubules. Protein synthesis governs traits like brown fur in this mouse. Genetic information is transcribed from DNA to mRNA and then translated into proteins for phenotypic expression. Protein synthesis is broken down into 2 steps: transcription and translation. It begins in the transcription of an mRNA strand inside the nucleus. The nucleotide sequence in the DNA is used to make a complementary sequence in mRNA. The RNA polymerase enzyme uses a single-stranded DNA to make mRNA. It works in the 5 to 3 direction and lays in the 3 to 5 direction. After the mRNA is completed, protective ends called the 5-methyl G-cap and the 3-poly-A tail are added to form the mature mRNA transcript, which is then ready to leave the nucleus. Translation occurs at the ribosomes in the cytoplasm. mRNA carries the code for building a protein to the ribosome. The ribosome reads the code and builds the protein using tRNA. tRNA carries amino acids to the ribosomes and lays them together. They read the mRNA by every 3 bases, which is called a codon. The tRNA lays the amino acids and connects it to the previous one before leaving the ribosome. The process is repeated until 1 amino acid is formed. Once these traits are coded for in protein synthesis, They get passed down to offspring in a branch of biology called genetics. Alleles are traits that come in alternate versions. An allele is a sequence in the DNA that codes for a certain trait. Some alleles are dominant, which means they mask other alleles which are recessive. For each trait, an individual inherits two alleles, one from each parent. How do people know which alleles each offspring will receive? Reginald Punnett was an English geneticist who developed the Punnett square. The Punnett square is a tool to help predict the probability of genotypes of offsprings given a cross between two parents using Mendel's law of segregation. Using the probabilities of Punnett squares, people can create a pedigree. A pedigree is a chart used to trace the phenotypes and genotypes in a family to determine whether people carry disorders or traits. Genetics is the study of how traits get passed down to offspring, and evolution is the process by which organelles change over time so their phenotypes are best suited to their environment. In the rock pocket mice population, there are two different colors of fur, light and dark. Living in the light-colored dirt, mice with light-colored fur blended in well, but mice with the dark fur did not. The dark-colored mice had a mutation in their DNA that already existed, giving them a disadvantage. The geography of the rock pocket mouse was a dry, desert climate with light-colored soil. However, there was a volcanic eruption and the lava flowed over the soil. When the lava cooled, it left black rock that matched the color of the black mice. The black-colored mice now had an advantage over the light-colored mice, and the allele frequency changed. Before the lava eruption, the recessive allele frequency was 84%, and the dominant allele frequency was 16%. 10 years after the eruption, the recessive allele frequency was 45%, and the dominant allele was 55%. Organisms can evolve over time to be best suited to their climate in a process called evolution, and ecology is the study of how these organisms relate to each other in an ecosystem, with hawks preying on mice. The white pocket mice had a competitive disadvantage after the eruption of the volcano. Their skin color was selectively chosen because it would be easily seen against the dark ground compared to the dark-colored mice. This is an example of disruptive selection. Over time, the white pocket mice population will slowly decrease as the population of the dark rock pocket mice increases. In conclusion, biology, or the study of life, includes intricate cellular processes that make up living and breathing organisms that interact with each other, pushing evolution through natural selection.